Hello students, today we are going to just see and solve the questions of botany of NEET 2021 held on September 12th this year. So let's now see question number 101. Plants follow different pathways in response to environment or phases of life to form different kinds of structure. This ability is called 1. Flexibility, 2. Plasticity, 3. Maturity, and 4. Elasticity. So the correct option is 2. Plasticity. So when the plants follow different pathway in response to environment or phases of life to form different kinds of structure, this ability is called plasticity. So a very good example of plasticity is heterophilia. So heterophilia is the presence of the different kinds of leaves, more than one type of leaf in the same plant. So heterophilia is found in many plants like cotton, larkspur, etc. So in the juvenile phase, they just coriander. So in juvenile phase, the leaves are small in order to withstand the brunt of wind, the force of wind. But as the plants just grow and they become a little strong, so their leaf just come to the full form, they just increase their surface area, as we can just see in the following figure. So here we can just see in figure A. Secondly, it is also found in buttercup, that is ranunculus, as we can just see in the second figure. So these are the filament-like leaves that are found in water habitat. Ranunculus is found in water, running water, and the leaves that are above water are normal. They are like normal simple leaves, but those leaves that are underwater, just facing the running water, so they just get filamentous in order to just survive, in order to just tolerate the force of running water, so that the plant doesn't get broken, the leaves do not get broken. So this is why this heterophilia is found in case of buttercup. So all these features just come under the character of plasticity. Let's now see question number 102. In spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? 1. Competitive release, 2. Mutualism, 3. Predation, and 4. Resource partitioning. The correct option is resource partitioning. Resource partitioning means just sharing the different components of the resource by virtually just drawing a line, but virtually just partitioning the resource and utilizing the different components of resource by the competing members of the species. So here, the correct option is resource partitioning. On the other hand, competitive release is the forceful expulsion of the weaker members, the weaker species members, when the arrival of some more dominant species just come in the habitat. So here, this is competitive release. Similarly, as we have just seen that when goat arrived in the Galapagos Island, so the tortoise had to just leave that habitat because they were more vigorous grazers. Mutualism is basically a mutually beneficial relationship between the two species. Very good examples are mycorrhiza and lichen. In lichen, we just see an alga and a fungus. They just come together and they form the lichen body to just benefit each other. Predation is the eating of one species by some other member of a species. So this is predation. So the correct option is a resource partitioning, by which, despite just belonging to the same niche, by resource partitioning, the two species just survive in the same niche. So let's now see question number 3. 103. Which of the following stage of meiosis involves division of centromere? 1. Metaphase 2. Second, anaphase 2. 3. Telophase 2. And 4. Metaphase 1. So the correct option is 2. Anaphase 2. Let's now just see the scenario in anaphase 2. First of all, in anaphase 2, the first let's see metaphase 2 and then anaphase 2. In metaphase 2, the chromosomes with the chromatids are arranged at the metaphase plate, equatorial plate, just as we see in mitotic metaphase. So the chromosomes are not paired, and the spindle fibers just come from the opposite poles and they get attached to the centromere. It is the centromere. And it is the chromosome with the two chromatids. It is the spindle fiber. So this is a metaphase 2 of meiosis 2. Now, as the anaphase 2 comes, the centromere is torn apart. It is just broken down, and this just follows the separation of the sister chromatids. These are the sister chromatids belonging to the same chromosome. So now, as anaphase 2 comes, the spindle fibers shorten. They just pull apart the sister chromatids, and this leads to a split of the centromere and subsequent movement of the sister chromatids to the opposite poles. So this is the anaphase 2. In telophase 2, these chromatids just get upon the poles. They just reach the poles, and this is why there's already a split of the centromere. Okay. In metaphase 1, the chromosomes in pair just come upon the equatorial plate like this. And subsequently, so this is metaphase 1, and subsequently the spindle fibers just pull apart the entire chromosomes, and there is no split of centromere. So the best answer is anaphase 2. So this is the correct answer. Let's now see the next question. Genera like Selaginella and Salvinia produce two kinds of spores. Such plants are known as 1. Heterosporous, 2. Homosporous, 3. Heterosporous, and 4. Homosporous. So the correct option is 3. Heterosporous. Heterospory is the phenomenon in which the two kinds of spores are produced by the same plant. One is the microspore and the other is the megaspore. Okay, so heterospory involves production of two types of spores. microspores and megaspores. Microspores gives rise to male gametophyte, whereas megaspores gives rise to female gametophyte. So microspore produces male gametophyte and megaspore produces a female gametophyte. It is noteworthy that microspore is named so because microspore is very small. It has got a very small amount of food, but it is produced in large number, and it is light, and it is dispersed to find out the megaspore that contains the female gametophyte and female gamete. So here megaspores are larger. They are produced in a small number, and they just develop the female gametophyte in which the archegonia and ova are produced in the lower plants. In higher plants like angiosperm, the archegonia are not found, but ova are present in the female gametophyte, that is the embryo sac. So here the phenomenon is heterosporous. I must just inform you that Selaginella and Salvinia, they are pteridophytes. Selaginella is non-fern pteridophyte and Salvinia is an aquatic fern. So let's now see question number 105. DNA stands on a gel stained with ethidium bromide when viewed under ultraviolet radiation appears as 1. Bright orange bands, 2. Dark red bands, 3. Bright blue bands, and 4. Yellow bands. So ethidium bromide is basically a dye. It is a fluorescent dye. And when gel electrophoresis takes place, it is the agarose gel. And when the different sizes of DNA are extracted in the form of band on the agarose gel in the process of gel electrophoresis. Gel electrophoresis. The bands of DNA are obtained, but they are not visible to the naked eye. They are not visible to the eyes. Okay. So suppose we are just performing DNA fingerprinting, and in this process, gel electrophoresis is made of the DNA. It is this technique is used anywhere: extraction of the plasmids from bacteria, separation from genome, bacterial genome, etc. So these bands are there according to their size. So these bands are treated with the ethidium bromide. The agarose gel is treated with ethidium bromide, and when ultraviolet rays are just focused on them, ultraviolet rays are incident. It is just falls upon the agarose gel. So these bands just shine as the bright orange bands, and even DNA can be extracted just locating those bands. So this is very useful. This staining is very useful in order to locate the DNA, and the DNA can be just extracted from the agarose gel, a process that is called elution. Okay, so the correct option is 1. Bright orange bands. Let's now see question number 106. Mutations in plant cells can be induced by 1. Infrared rays, 2. Gamma rays, 3. G18, and 4. Kinetin. So the correct option is gamma rays. Infrared rays are low energy rays. No doubt they are the physical radiations, but they are not visible. They are they have got a very long wavelength. They are rather just felt in the form of heat, and they are not capable to just cause mutation in the plant cells. Gamma radiations just come from the radioactive substances. Alpha rays, beta rays, and gamma rays are the radioactive radiations from the radioactive substances like cobalt-60. And gamma rays are just bombarded upon the seeds, etc., in the gamma gardens of the atomic agencies, from where, which just get the plants from the plant breeding centers, so that gamma radiations can be just put upon them, and mutations can be just caused in order to develop the mutant varieties that may be of some use. Okay, so gamma rays is the correct option. G18 and kinetin are actually cytokinins. Both G18 and kinetin. Kinetin is the first cytokinin to have been discovered, and G18 is a natural cytokinin that is present in the maize as also coconut water. So they are not capable of just causing mutation. So the correct option is gamma rays. So let's now see question number 107. When gene targeting involving gene amplification is attempted in an individual tissue to treat disease, it is known as first, gene therapy; second, molecular diagnosis; third, safety testing; and fourth, biopiracy. So the correct option is gene therapy. Some heavy words have been used in this question. First of all, when gene targeting means delivering the gene to a particular location. For example, a person who is suffering from some genetic disease. So if a healthy gene is delivered into its body, so it is gene targeting. Gene amplification means just expression of the gene in amplified manner at a much larger scale. And for this, a repetitive DNA is a repetitive gene is introduced, a gene in which repetitive sequences are introduced, or a number of copies of the genes are just delivered. Okay, so this technique is used to just deliver the healthy gene, a number of copies of the healthy genes into the person who is suffering from some genetic disease. For example, so this is called gene therapy. For example, a girl suffering from SCID. SCID stands for severe combined immunodeficiency syndrome. So a girl who just suffered from SCID was just given the dose of the healthy gene of ADA. Actually, this disease occurs due to deficiency of adenosine deaminase. Adenosine deaminase enzyme. So due to defect in the gene that makes this enzyme, the functional adenosine deaminase enzyme is not available, and this just prevents the maturation of WBC, and this just leads to the non-functioning of the immune system. So the girl who suffered from severe combined immunodeficiency syndrome was given the healthy copies of the ADA gene through the retroviruses, and the girl just got cured. However, she had to just take this gene introduced into her bone marrow almost every 4 years. Okay, so this was an attempt. It was a successful attempt of gene therapy in the human being. So here the correct option is the gene therapy. Molecular diagnosis is the use of the certain molecule to diagnose certain disorders or germs, etc. So PCR and ELISA technique are included in the molecular diagnosis. Safety testing is the testing of the drugs for the safety in the human beings. And biopiracy is actually the illegal claim of certain multinational companies upon the biological resources of the third world countries. So the correct answer is gene therapy. So let's now see question number 108. When the centromere is situated in the middle of the two equal arms of chromosomes, the chromosome is referred to as 1. Telocentric, 2. Submetacentric, 3. Acrocentric, and 4. Metacentric. So the correct option is 4. Metacentric. So if we just see these figures, it will be more clear. Here we just see that the centromere is present in between the two arms. The two arms are equal in length, and the centromere is just in the middle of the two arms, and this condition is called the metacentric condition. On the other hand, if one arm is slightly smaller than the other arm and the centromere is not in the middle, rather slightly away from the middle, so this condition is called submetacentric. As we just see in the case of X chromosomes. So it is the submetacentric condition. So the centromere is slightly away from the middle of the chromosome, and the two arms are unequal. When the centromere is near one end, it is this. So one arm is very long and the other arm is very small. So this condition is called the acrocentric condition, as we just see in case of Y chromosome. So this condition is called acrocentric condition. And lastly, if the centromere is at just one end and there's only one arm, the other arm almost too diminished to be noticed. So this condition is called telocentric condition, as we can just see here. So this is the centromere. Centromere is at one end, and this is the only arm, very long arm. So this condition is called telocentric condition. Okay, so this can be just remembered by these shapes. It is V, the centromere in the middle. It is L. It is J. And it is I. So V corresponds to metacentric, L corresponds to submetacentric, J corresponds to acrocentric, and I corresponds to telocentric. Okay, so let's now see the next question, question number 109. So here, this is the question about the periderm and bark. Okay, so periderm is a protective layer that is formed by the cork cambium, and it has got the three components. This is to protect the plant once the secondary growth takes place, and the permanent tissues like epidermis and cortex just break down, and the inner parts of the plant just faces the risk of getting exposed to the germs and losing water. So periderm is formed by the plant beforehand. So here, one is the cork cambium. Cork cambium is called phyllogen. So it just forms towards the inside of the plant, the phylloderm, the living cells, and phylloderm is called the secondary cortex. It's called the secondary cortex. And outside, it just forms a layer that is called cork, also known as phylum. So cork cells soon just form the secondary cell wall made of suberin, and due to heavy suberin deposition, they just get dead, and they play a very important role in the formation of the bark. Bark is a loose term that includes the dead cells like cork, as also some living cells. Okay, so bark has got certain pores which are known as lenticels. Lenticels are the minute pores in the bark. The function of the lenticel is the gaseous exchange between atmosphere and the stem. So here, first of all, in context of these facts, let's now just see the columns, list 1 and list 2. So lenticels is meant for exchange of gases. Yeah. Cork cambium is phyllogen. Further, secondary cortex is phylloderm. Okay. And cork corresponds to suberin deposition. Okay. So the correct option is A1. A3, sorry. So A3, B1, C, 4, and D, 2. So the correct option is 1. Okay. So let's now see the next question, question number 110. Which of the following is an incorrect statement? Microbodies are present both in plants and animals. It is correct. The perinucleate space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Okay, let's now just see it. Nuclear pores act as a passage for proteins and RNA molecule in both directions between nucleus and cytoplasm. Okay. Mature sieve tube elements possesses a conspicuous nucleus and usual cytoplasmic organelles. So this is actually the wrong option. This is incorrect statement. It is because the sieve tube elements found in the angiosperms, so they do not have the nucleus, but they are the living cells, and in order to maintain their metabolism, control the activity of the sieve tube elements, there is a companion cell. Okay, so if we just see the structure of a sieve tube, I must just mention you that there are two types of sieve elements. One are the sieve tube and the other are the sieve cells. Sieve cells are found. Sieve cells are nucleate. Just focus here, cells, not tube. So sieve cells are nucleate, found in pteridophytes and gymnosperms. But absent in the flowering plants, in angiosperms. On the contrary, the sieve tube, it is not sieve cells, rather it is sieve tube. So sieve tube are living cells, but despite that, they lack a well-defined nucleus. They actually lack the nucleus. Okay, nucleus degenerates as the sieve tube matures. So here, it is a sieve plate on the two ends. There is a central large vacuole, and many other vital organelles are absent here. But it is accompanied by a companion cell. So this is the sieve tube. It is the sieve plate with pores. It is a sac vacuole. And it is a companion cell, the nucleus of which controls. Companion cell is the nucleate. And this nucleus also controls the activity of the sieve tube. So here, the mature sieve tube elements do not have any conspicuous nucleus, and usual cytoplasmic cell organelles are also absent, so that the flow of the food becomes more fast, it becomes more efficient. On the other hand, microbodies like peroxisomes, lysosomes, peroxisomes, glaucosomes, etc. So they are present in plants and animals as well. Plants do not have the lysosome, but they have got the peroxisomes and glaucosomes, and animal cells have got the peroxisomes and lysosomes. So they are present in both plants and animals. The perinucleate space forms a barrier between the materials present inside the nucleus and that of cytoplasm. In a nucleus, the space in between the two nuclear membranes is called perinucleate space. So this is the nucleus, and it is a nucleolus, and it is a chromatin material. It is a chromatin fibers, an intricate mass of chromatin fibers. So these are nuclear membranes. It is the space that is known as the perinuclear space. These are nuclear pores. And ribosomes just a dot upon the surface of the outer surface of the outer nuclear membrane, and it is a rough endoplasmic reticulum that projects out from the outer nuclear membrane. So here, these are the nuclear pores. Through which there is exchange of substances between the nucleoplasm and the cytoplasm. There is a nucleoplasm, and it is the rough endoplasmic reticulum. It is the nucleolus, and it is a chromatin fiber. So in this light, when we just see that the two options, the perinucleate space forms a barrier between the materials present inside the nucleus and that of cytoplasm. So this is the correct option. Nuclear pore acts as a passenger for protein and RNA molecule in both directions between nucleus and cytoplasm. It is again a correct option because many proteins just enter the nucleus formed upon the ribosome in cytoplasm. And on the other hand, after transcription, when the RNA molecules are formed from DNA by RNA polymerase, so RNA just moves over to cytoplasm and there they have to just participate in the formation of the proteins. So option 3 is also correct. So it is option 4 that is the correct option because this is the incorrect statement. Let's now see question number 111. Gemmi are present in pteridophytes, some gymnosperms, some liverworts, and 4 mosses. So the correct option is some liverworts. Example given, Markantia. Markantia is a liverwort, a bryophyte. Okay, mosses are also bryophyte, but they don't have the gemmi. So gemmi are actually the ear-shaped flattened structures like this with a stalk, and numerous such gemmi are found on the cup-like structure on the surface of Markantia, which are known as the gemma cup. So many such gemmi are there. And many other filaments are there. So these are gemmi. And if we just see a gemma from side, we just find that it is a flattened structure. Okay, so it is a gemma. So gemma is an adventitious structure. It is meant for vegetative propagation. And as it breaks down and falls upon the ground, so it just gives rise to the new plant. Okay, so this is a gemma, and it is a gemma cup, which is found on the surface of Markantia. So such cup-shaped structures are there, which are known as gemma cup, and these gemma cups just contain a number of these gemmi, which just falls upon the ground, and ultimately they give rise to the new liverwort plant. So the correct option is C. That is the 3. So let's now see question number 112. Which of the following plants is the monoecious? 1, Cara. 2, Markantia polymorpha. 3, Cycas sarcinalis, and 4, Carca papaya. So the correct option is 1, Cara. Actually, what we see in the Cara plant is that both the male and the female reproductive parts, known as the oogonia and antheridia, are present on the same plant. So here, this is the structure, the rear jacketed structure with the ovum is present here, and on the lower side there occurs the antheridium. So it is the oogonium, and this is the ovum. Oogonium is also called nucleus, and this is the entheridium that produces the male gametes, also called globule. So both are present on the same plant, the nucleus on the higher position and the globule on the lower position, and these are branches. It is the main axis. So in this way, Kara is a monoecious. Monoecious condition is that condition in which both the male and female structure are found on the same plant. However, Markantia is a dioecious in which the male and female plants are separate. A dioecious condition is one in which the male and female plants are separate. The male plant contains the male reproductive structures and the female plant contains the female reproductive structures. So Markantia is dioecious. Carica papaya is a dioecious. It is the dioecious angiosperm. Markantia is a dioecious bryophyte, dioecious liverwort, and Cycas is a dioecious gymnosperm. So the rest three are dioecious, whereas Kara is a monoecious. Okay. So let's now see question number 113. Which of the following is not an application of PCR, polymerase chain reaction? 1. Gene amplification. 2. Purification of isolated proteins. 3. Detection of gene mutation. And 4. Molecular diagnosis. Okay. First of all, what is polymerase chain reaction? Polymerase chain reaction is the technique in which a single DNA can be multiplied, it can be just copied, and within hours, billions of copies can be formed. Okay, so this is a very good way of gene amplification. That is, the gene is multiplied so that its expression can occur on the larger scale. So this is a very good example of gene amplification. Secondly, polymerase chain reaction is also a way of detection of gene mutation, because when the gene just comes in bulk, it just when billions of genes are there, so it becomes very easy to find out any genetic mutation, any gene mutation. Okay, so this is helpful in gene detection of gene mutation as well. Molecular diagnosis is a very important application of polymerase chain reaction, in which the genome of a person, the genome of the particular organism is multiplied, and the content, if the content also just shows the genome of some germ as well, so this just leads to detection of some disease as well. So this is a very efficient and very sensitive way of molecular diagnosis. However, purification of isolated protein is completely out of the application of PCR. Actually, PCR is never used for purification of isolated protein, because PCR deals with the DNA and not with the protein. So here the correct option is the purification of isolated protein. Okay, so these three, gene amplification, detection of gene mutation, and molecular diagnosis, are the functions of polymerase chain reaction. So let's now see question number 114. The production of gametes by the parents, formation of zygote, the F1 and F2 plants can be understood from a diagram called Punched square, 2nd, Punnett square, 3, net square, and 4, bullet square. So the correct option is 2, Punnett square. The British scientist Punnett just devised the box-like representation, the box-like chart upon which on the two ends there are the locations for the gametes, and subsequently in the middle boxes contain the genotypes representing the organisms with certain genotypes. Okay, so I'm just giving one example. Suppose there's a cross in between capital T small t into capital T small t. There's a dominant T and a recessive T and dominant T recessive T. One is a male and the other is a female. So they just make their gametes, and this can be represented, this cross can be represented by this box-like structure. So here, this part represents the male gamete and this part represents the female gamete. So on one side, we just show the male gamete and the other side, the female gamete. So here, let's just take it female and it is as male. So the gametes of male are shown here and the gametes of female are shown here. So here, capital T, small t, dominant and recessive T, dominant T and recessive T. So these are these represent the male gamete and these represent the female gamete. And here we may just fill the larger boxes. So dominant T, dominant T, dominant T, recessive T, dominant T, recessive T, and recessive T, recessive T. So it is a pure tall. It is pure tall. Hybrid tall. It is hybrid tall. And it is pure dwarf. So here, in this way, we may just represent the F1, F2 generations very conveniently. Okay. So this also just minimizes the chances of mistake in the larger process, particularly dihybrid process. So this representation just gives more accuracy in order to show the F2 and F1 and F2 generations. Okay, so the correct option is Punnett square. So let's now see question number 115. A typical angiosperm embryo sac at maturity is 7 nucleate and 8 celled, 7 nucleate and 7 celled, 8 nucleate and 8 celled, and 8 nucleate and 7 celled. So the correct option is 8 nucleate and 7 celled. So let's just see an embryo sac, which is actually the female gametophyte found inside the ovule. The embryo sac that is formed from megaspore formed by the megaspore mother cell, a part of nucellus. So in an embryo sac, this is the ovum. Here the two synergids. These are the two synergids. And there are two polar nuclei of the central cell. And here, the antipodals. So here we see that these are antipodals. These are antipodals, which are three. There's a central cell. It is a central cell which has a large vacuole, subvacuole. These are the two polar nuclei of the central cell. And it is the ovum. And these are the two synergids. So here we find that there are two synergids, one ovum, one central cell. But if we just make a count of the nucleus, we find that antipodals have altogether three nuclei. But the central cell has got two nuclei, two polar nuclei. The ovum has got one nucleus. And synergids have got the two nuclei. So if we just tally the number of the cells and the nucleus, we find that even while there are 7 cells, but the central cell being binucleate, so it just raises the number of nucleus to 8. So the correct option is 8 nucleate and 7 cells. That's the fourth option. Okay, so let's now see the next question, question number 116. The term used for transfer of pollen grains from anther of one plant to the stigma of a different plant, which during pollination brings genetically different types of pollen grains to a stigma, is called A. Geitonogamy, 2. Chasmogamy, 3. Cleistogamy, and 4. Genogamy. So the correct option is 4. Genogamy. Genogamy is another name of cross-pollination. Okay, so cross-pollination is or genogamy is a phenomenon in which the pollen from the anther of the flower of one plant just goes through some agencies, abiotic or biotic, to the stigma of the pistil of the flower of some other plant. Okay, so nature just favors it because it brings about variation faster compared to the self-pollination. Geitonogamy is the pollination in which the pollen grains just pass from a flower of a plant to the other flower of the same plant. Okay, so geitonogamy is between flowers of the same, between two flowers of the same plant. Of the same plant. Okay. Chasmogamy is the pollination within the same flower when the flower is open. Within the same flower, when the flower opens. Chasmos means open. Cleistogamy is within the same flower, but the flower never opens, it remains closed. Within a flower, which remains closed. Okay, so chasmogamy as found in Mirabilis in case of Catharanthus. Clistogamy, very good example is Camelina bengalensis. Geitonogamy found in the case of cucurbits, and genogamy, which is found in salvia, in yucca, where special insect adaptations are there and flower adaptations are there for this purpose. Okay, or where the birds pollinate, or the wind pollination is there, like the monocots like maize, wheat, etc. So genogamy takes place, or cross-pollination takes place. So the correct option is genogamy or cross-pollination. Okay, so let's now see question number 117. The factor that lends to founder effect in a population is 1. Genetic recombination, 2. Mutation, 3. Genetic drift, and 4. Natural selection. So the correct option is the genetic drift. Genetic drift a term given by Sewell Wright. So here just given the term genetic drift. So genetic drift is the sudden change in the frequency of genes. In the frequency of genes is called genetic drift. I must better use the term alleles. Okay, so this is called a genetic drift. So genetic drift, you have just studied genetic drift in class 10th as well, where you have just gone through the story of the beetles, the red beetles which were in majority, and blue beetles, which beetles are a small, very tiny insects. So the blue beetles are also there. So a population of beetle contains both a red beetle and a blue beetle. And here, so red beetle population is very large and blue beetle is a small, but suddenly an elephant just passes and it just puts its one foot upon the population, and most of the red beetles are killed, and it is now the blue beetles that are in majority. So this is a sudden change in the frequency of the alleles. Okay, so this is actually the bottleneck effect, because the population suddenly just drops. And now the new population, the allelic frequency of the new population decides the future generations and the future gene frequency and subsequent direction of the evolution. However, in founder's effect, a population just goes from one place to the other place. Okay, when a small part of a population just migrates from one habitat to the other habitat, and where any population of that species lives. Okay, so this also changes the allelic frequency, because it is the allelic frequency of the migrating part of the population that now just decides the allelic frequency of the newly populated land. Okay, newly populated habitat. So this is called founder's effect. Suppose, I'm just giving you one example. In the mainland America, 90% are white. Yes. So 90% are white and 10% is Negro population. Now suppose an island which is unpopulated, So some migration takes place, and this migration just it is in the ratio of 80% Negroes and 10% white. So migrate, founding the population there. Founding means laying foundation. The population. So subsequently, that island will now have, when it gets populated, so subsequently, this island now contains 80% Negroes and 10% and 20% white. So here the allelic frequency has changed, and if there is no intermixing of the populations of that island and the mainland USA, so obviously this island will just evolve. The population of this island will evolve with respect to that particular gene in altogether different direction. So here again genetic drift is playing its role, but it is due to the foundation of a new population. So this is called the founder's effect. Okay, so it is also an effective way of the allelic frequency change and subsequent direction determination of evolution. So let's now see question number 118. Which of the following are not secondary metabolites in plants? First, amino acids and glucose. Second, vinblastine, curcumin. 3, rubber, gums. And 4, morphine and codeine. So here we find, first of all, let's see what the secondary metabolites are. Secondary metabolites are some byproducts, some products found in the organisms that do not play the role in the main metabolism. In major metabolism, they do not play any major role. Okay, so here in these options, the correct option is 1. What we have to find? Not. They are not the secondary metabolites. So amino acids and glucose are major metabolites. They are the primary metabolites that participate in the major metabolism, like formation of the proteins by amino acids, formation of starch, glycogen, etc. Breakdown of glucose is the main phenomenon in respiration. Okay, so option 1 just include those which are not the secondary metabolites, whereas the other 3, that the vinblastine and curcumin, rubber and gums, morphine and codeine, they are the secondary metabolites. So correct option is 1, amino acids and glucose. Let's now see question number 119. The plant hormone used to destroy weeds in a field is NAA, 2, 2,4-D, 3, IBA, and 4, IAA. So the correct option is 2, 2,4-D. All these are auxins. So here it is the naphthalene acid. NAA stands for naphthalene acid. Two 4D stands for 2,4 dichloro phenoxyacetic acid. IBA stands for indole butyric acid. and IAA stands for indole acetic acid. Indole 3 acetic acid. So 2,4-D is actually a synthetic oxygen, and it is used as the weedicides. Okay, so those chemicals that are meant to kill the weeds in a field of crops, so they are called weedicides. So oxygen just functions as a weedicide, and here the synthetic oxygen 2,4-D is used in the fields in order to destroy the weeds. So the correct option is 2,4-D, synthetic oxygen. Let's now see question number 120. Amensalism can be represented as 1. Species A plus, species B plus, species A minus, species B minus. 3. Species A plus, species B minus 0. And 4. Species A minus and species B 0. So the correct option is 4. Amensalism is a negative kind of relationship. If we just see all the 4, so first of all, we see 1, species A is benefited and species B is also benefited. So this is mutualism. So the first is the case of mutualism. The second is species A is harmed, species B is also harmed. So this is the twin negative relationship that we just find in competition. These are actually the biotic interaction just occurring in an ecosystem. So here it is competition. When one species is benefited and the other species is neither harmed nor benefited, so this is commensalism. This is called commensalism. Okay. However, if a species is harmed and the other species is not benefited, so it is called amensalism. So this is amensalism. Okay, so the correct option is 4, amensalism. A very good example of amensalism is antibiotic production, antibiosis. Okay, this is why amensalism is also called antibiosis. Okay, so the production of antibiotics by certain organism in order to kill the other is one very good example of amensalism. For example, the filamentous bacteria just produce many antibiotics, and so do the fungi like penicillium. So penicillium notatum and Penicillium chrysogenum both produce penicillin, whereas the Streptomyces venezuelae produces chloramphenicol. Also known as chloromycetin. So all these are the examples of antibiosis or amensalism. Let's now see the next question. Question number 121. Which of the following algae contains mannitol as a reserve food material? 1. Gracilaria, 2. Volvox, 3. Ulothrix, and 4. Ectocarpus. So the correct option is 4. Ectocarpus. Gracilaria is basically a red alga, and it is the source of agar. So it just contains the rhodophycin starch. It contains rhodophycin starch as a reserve food. Volvox is a green alga, so is the Ulothrix. So both Volvox and the Ulothrix, they just contain a starch. Both of them just contain a starch as a reserve food, whereas Ectocarpus is a brown alga. Gracilaria is a red alga. Volvox is a green alga. Ulothrix is also a green alga. And Ectocarpus is a brown alga. So brown alga just contain there is a food in the form of laminarin and mannitol. Okay, so the correct option is D, Ectocarpus. Okay, so let's now see question number 122. The amount of nutrients such as carbon, nitrogen, phosphorus, and calcium present in the soil at any given time is referred to as climax community, a standing state, a standing crop, and 4. Climax. So let's see the question. The amount of nutrients such as carbon, nitrogen, phosphorus, and calcium present in the soil at any given time is referred to as 1. Climax community, 2. A standing, 3. A standing crop, and 4. Climax. So here the correct option is 2. A standing state. A standing state is the presence of the inorganic components in an ecosystem at any given time. So carbon, the contents of carbon, nitrogen, phosphorus, and calcium present in the soil at any given time is referred to as a standing state. Climax community is a final community, it is a final group of population that live when the ecological succession leaves its final stage, and it is usually a forest. A standing crop is the total biomass that is present in an environment, in an ecosystem at a given time. And climax is actually the last stage of the succession, ecological succession. So here the correct option is a standing state. So let's now see question number 123. Which of the following algae produce carrageen? So 1 is brown algae, the 2nd is red algae, the 3 is blue-green algae, and 4, the green algae. So the correct option is 2, red algae. Carrageen is basically a component of the cell wall of red algae. So it is a phycocolloid. Like agar, that is also found in the red algae. So it is used in many, you can say, applications for the manufacture of ice cream as also paint, as also the shoe polishes, etc. Brown algae just give the algin obtained from alginic acid. Blue-green algae and green algae, they are not the source of carrageenan. So the correct option is 2, red algae. Let's now see the next question.
Recording in progress.
So in the equation, GPP minus R is equal to NPP. R represents first, retardation factor, 2, environmental factor, 3, respiration losses, and 4, radiant energy. So the correct option is respiration losses. So what is GPP? GPP stands for gross primary productivity. Okay, so it is the total amount of the food that the plant form. Okay, now plant in an ecosystem just forms. Now R is the respiratory loss. Loss of the food through respiration. And NPP stands for net primary productivity. Okay, so here when we just deduct, when we subtract the respiratory loss from the gross primary productivity, that is when we just subtract the food that has been consumed in respiration, so we get the net primary productivity. So here the correct option is 3, respiration losses. Let's now see the next question, question number 125. The site of perception of light in plants during photoperiodism is the 1, stem, 2, axillary bud, 3, leaf, and 4, shoot apex. So the correct option is 3, leaf. Photoperiodism is a phenomenon in which it is the duration of the light that is important for flowering. Okay, so the role of the duration of light in flowering is called photoperiodism. Okay, so the light is perceived, the light is just received, the part of the plant that is sensitive to light in order that photoperiodism can take place, flowering can take place. So it is leaf. Okay, leaves are so effective, they are so sensitive that even a single leaf present on the plant can just take up the input of the duration of light. It may just perceive the light. Okay, and this perception just causes the conversion of the shoot mestem into the floral mestem. Okay, and the hormone Florigen is formed in response, and the photoperiodism takes place. Photoperiodism is a phenomenon in which light plays a major role in the developmental activity of the plant, and such phenomena are collectively called photomorphogenesis. Okay, so photoperiodism is a photomorphogenetic process in which even a flash of light can bring about the difference. So it is the leaf that's the correct option. Let's now see question number 126. Diadelphous stamens are found in 1, citrus, 2, pea, 3, China rose and citrus, and 4, China rose. So the correct option is 2, pea. Actually, diadelphous condition is there in which the stamens are present in two groups. So when diadelphous condition involves presence of the stamens into two groups. Okay, so here, as we just see, that P is actually the member of family Fabaceae. It is a member of family Fabaceae. And stamens are present in a family Fabaceae in a group of 9 and 1. Okay, so here in case of members of family Fabaceae, stamens are grouped into 9 stamens on one side and one stamen on the other. So here we can just see this condition in the following figure. So we find that 9 stamens are here that are just cohesive to each other, and one stamen just stands apart from the rest 9. So this condition is found in family Fabaceae, including the P, that is Pisum sativum. Okay, so this P, the scientific name of P is Pisum sativum. So this condition is found. So the correct option is option 2. Let's now see question number 127, a matching column type of question. So here, let's see column 1. Cells with active cell division capacity. Tissues having all cells similar in structure and functions. Tissue having different types of cells, and dead cells with highly thickened walls and narrow lumen. Now, the list 2 contains the list vascular tissue, meristematic tissue, sclerates, simple tissue, etc. So, first, let's see cells with active cell division capacity. So, it is the meristematic cell. It is this. Tissue having all cells similar in structure and function. So it is the simple tissue. Okay. Tissues having different types of cells. So they are vascular tissue because they just make the complex tissue, and they have more than one type of cells, mesenchyma, parenchyma, etc. Now, dead cells with highly thickened walls and narrow lumen. So this is the sclerate. That's a part of xylem. Okay. So the correct option, now let's see the options. So here A for 2. So here it is 2. B for 4. It is B for 4. C for 1. It is correct. And D for 3. So the correct option is the 4. Okay. So let's now move over to the next question. Question number 128. match list 1 with list 2. Cristae, thylakoid, centromere, cisternae. And let's now see primary construction in chromosome, disc-shaped sacs in Golgi apparatus, infoldings in mitochondria, and flattened membranous sacs in stroma of plastids. Okay, so first let's see cristae. Cristae are actually the infolding in mitochondria. They are the infolding of the inner membrane of mitochondria. Okay, so it is the mitochondria. It is the outer membrane, and the inner membrane just remains folded into the mitochondrial matrix in such a way, and these in projections are called cristae. So these are cristae. Second, thylakoids. Thylakoids are actually flattened membranous sac in the stroma of plastids. So thylakoids are flattened membranous sacs in stroma of plastids. So thylakoids are such hollow coin-like structure that make the grana, and they are present inside the stroma of the chloroplast. Okay. Okay, so these are thylakoids. These are thylakoids. Now centromere. Centromere is a primary construction in chromosome. So here it just goes with 1. So if we just see a chromosome, so we find an area that holds the 2 chromatids, and even while the chromatids are not formed, it just holds the 2 arms. So this area is called centromere. And the cisternae are the disc-shaped sacs in Golgi apparatus. So it just goes with 2. So cisternae are such structures like this. It is, these are cisternae of Golgi body. So let's now just find the options. So A just tallies with 3. So here A tallies with 3, B tallies with 4, C tallies with 1, and D tallies with 2. Okay, so the correct option is 2. So let's now just switch over to the next question. Question number 129. Which of the following statements is not correct? First, pyramid of biomass in C is generally upright. Second, pyramid of energy is always upright. Pyramid of number in a grassland ecosystem is upright. Okay. And pyramid of biomass in C is generally inverted. So the correct option is 1. Actually, the pyramid of the biomass in C is generally inverted. Okay, it is because the phytoplanktons, which are the primary producers there, so they have a very high rate of reproduction, and they just continuously just feed over the upper trophic levels, whereas the members of upper trophic levels have got the longer lifespan, and they just keep accumulating the food obtained from the lower trophic levels, and therefore their biomass just becomes larger than the lower trophic level. So here, such kind of structure is found in the sea. Okay, so the primary producers are less in biomass because they are consumed, but at the same time, since their rate of reproduction is very high, so they just keep feeding the upper trophic levels. Okay, so it is the producers. It is the primary consumer, the shellfish, etc., the molluscans that feed upon the phytoplanktons. and seaweeds, and it is the fish that just feed upon the molluscans. Okay, it is the second. They are the secondary consumers. Okay, so option 1 is the correct option because it is not correct. It is the statement is not correct. So let's now see question number 130. Complete the flow chart on central dogma. So here the central dogma given by FSC Crick is all about the flow of information. So here we find that there are 3 points, A, B, and C. So DNA, being autocatalytic molecule, undergoes DNA replication, and the dependent DNA formation takes place, which is known as DNA replication. The second one is a DNA dependent RNA formation, that is called transcription. That's a B. And from mRNA, the formation of protein takes place on the ribosomes, a process that is known as translation. So here, the A is the replication. So we have got 2 options in which replication is starting. The second one is the transcription. So both 2 and 4 have got the transcription as B. Now the third one is a translation. Now here, it is the second option that contains a translation, whereas the fourth option is a transduction, which is not actually the process here involved here. And the fourth one is protein. So therefore, the correct option is 2. Replication, transcription, translation, and protein. Transduction is actually the transfer of a part of the genome of bacteria from one bacteria to the other bacteria by some temperate phage virus. So the option 2 is correct, whereas option 4 is wrong. So let's now see question number 131. The first stable product of carbon dioxide fixation in sorghum is 1. Oxaloacetic acid, 2. Succinic acid, 3. Phosphoglyceric acid, and 4. Pyruvic acid. So the correct option is oxaloacetic acid. Actually, sorghum is a C4 plant. Sorghum is a C4 plant. And we know that the first stable product of the primary fixation of carbon dioxide in the C4 plant is the oxaloacetic acid. So here, phosphoenol pyruvic acid acid, that is known as PP. So it just combines with the carbon dioxide and water. And one inorganic phosphate is given out in presence of enzyme PP carboxylase or peptase. And the first stable product, oxaloacetic acid, is formed. Which is actually a 4-carbon compound. Okay, whereas it is a 3-carbon compound. So here it is this is why it is called the C4 cycle. So let's now see the next question. match list 1 with list 2. So here are some processes related to the tissue culture, protoplast fusion, plant tissue culture, mestem culture, micropropagation. And the options are totipotency, palmetto, soma clones, and virus-free plants. So first of all, let's see that protoplast fusion is responsible for a hybrid like palmetto. Palmetto is formed by a combination of the cells of potato and tomato. Okay, so it is a hybrid prepared from potato and tomato. The plant tissue culture is actually the result of totipotency. Just 1 minute. Micropropagation is and mestem culture. Mestem culture is responsible for virus-free plants. Okay, and plant tissue culture just leads to the so plant tissue culture is attributed to totipotency. Okay, and micropropagation is involved. It just leads to formation of soma clones. So protoplast fusion is a technique in which the two cells are first of all made naked. Two plant cells are made naked with the use of the enzymes like cellulase and pectinase, and subsequently with the help of the substances like polyethylene glycol. The cells are made to fuse, and when we have to just fuse the cells of the two plants in order to just raise the hybrid, so we just put the naked cells of both the plants in a particular medium, and we just put polyethylene glycol that makes pores in the plasma membrane of the cells of both the plants, and this just ultimately leads to fusion of the cells of both the plants and the production of the hybrid. So palmetto is such a hybrid that has been prepared by the protoplast fusion of both potato and tomato cells. Plant tissue culture is the raising of the entire plant, starting either from the explant, a very small portion of tissue taken from the plant, or even a single cell, that the protoplast culture. So this is due to the ability of the plant cell, due to the special ability of every plant cell to develop into the complete plant. So this ability is called totipotency. Mestem culture is preferred, first of all, because mestematic tissue is already in a dividing condition, and they are easy to just easy to grow. And secondly, mestems are actually virus-free parts of the plant. Mestem cells dividing very fast, and the virus life cycle not coping up with that pace. So here, mestem culture just leads to virus-free plant. So this is another reason why mestems are preferred for the plant tissue culture. Micropropagation, a technique in which the very small part of the plant, so this just leads to soma clones. Soma clones are somatic clones. A clone is a population that is raised asexually from a single parent. Okay, so when a sexual reproduction takes place or vegetative propagation takes place, so there occurs the production of the asexually raised population that is called the clone. So here the somatic soma clones are the populations. It is the population that is produced by micropropagation of either a tissue or a single cell. So here the correct option, if we just see here, so the correct option is 1. Okay. So let's now see question number 133. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out first DNA, 2 histone, 3 polysaccharides, and 4 RNA. So the correct answer is the DNA. So when we just digest the different components of the cell, we break the cell, we digest the different components of the cell by using the enzymes to digest all the components except DNA. So lastly, we just get a suspension in which the different components are dissolved except DNA. So we just add chilled ethanol and we just take out the DNA. Okay, so a process that is known as spooling. So here it is the DNA that is just taken out, and it is very important in genetic engineering. Okay, so here let's now just see the next question. Question number 134. Which of the following is a correct sequence of steps in a PCR, polymerase chain reaction? So our options are denaturation, extension, annealing. Extension, denaturation, annealing. Annealing, denaturation, extension. And the fourth option is denaturation, annealing, and extension. So the correct option is 4. Denaturation, annealing, and extension. What is denaturation? Denaturation is the separation of the two strands. Separation of the two strands of DNA by heating, annealing, or renaturation of bringing union of the two strands. Okay, and extension is actually the formation of the new strands. Okay, so the correct option is 4. That is denaturation, annealing, and extension. Okay, so let's now see question number 135. Cohesion, adhesion, surface tension, guttation. So here the options are more attraction in liquid phase, B, among water molecule, water loose in a liquid phase, and D, guttation, attraction towards a polar surface. Okay. So the correct option is 4. How? Let's now just see. So cohesion is actually the property of sticking together of the similar molecules. So this is found among the water molecules. Water molecules have a tendency to stick together, and this helps water to form a continuous column inside xylem. Okay. Now, adhesion is the attraction towards the polar surfaces. Adhesion is actually the sticking of the two dissimilar molecules. Okay, so water also just moistens. Water also sticks to other molecule, and particularly the molecule that contains the polar surface that has got the slightly you can say slight charges upon that. Okay, so it also just sticks to that surface. So B is for 4, and C is 1, more attraction in liquid phase. Okay, so that is surface tension, and guttation is actually water loss in liquid phase. So guttation is actually the loss of water in the form of water droplets from the margins of the leaves. Okay, so here the correct option is 4. Okay, so let's now see the next question. So here section B has started. DNA fingerprinting involves identifying differences in some specific region in DNA sequence called as repetitive DNA, single nucleotides, polymorphic DNA, and satellite DNA. So here the correct option is repetitive DNA. Let's just discuss one by one. First of all, let's see what is DNA fingerprinting. DNA fingerprinting is a technique by which we just match the DNA of the two different sources. Okay, and here we don't match the entire DNA, rather we just match the presence of the specific sequences known as VNTR, that the variable number tandem repeats, or VNTR, also known as mini satellites. Okay, mini satellites are basically the repetitive sequences of of 11 to 60 base pairs. 11 to 60 base pairs. So they are called the mini satellite DNA, which is actually the repetitive DNA. Satellite DNA is a term that is used for the sequences bigger than 60 base pair. Okay, so single nucleotide is the wrong answer. Polymorphic DNA is again not a convincing answer. It is not a correct answer. Satellite DNA is not a correct answer because it is longer than 60 base pair in length. Okay, so this is why the best answer, the correct answer is a repetitive DNA. That is the good example of which is a mini satellite, which is a repetitive DNA. Okay, so these DNA mini satellites are actually repeated continuously. Tandem is continuous repeat, and they just vary in number on different chromosome of a person. And there are a number of types of such mini satellites, okay, such VNTRs, and it is the matching of the VNTR that we just do in DNA fingerprinting. Okay, so here let's now see question number 137. Which of the following statements is incorrect? In electron transport system, an electron transport chain. One molecule of NADH plus H plus gives rise to 2 ATP molecule, and one FADH2 gives rise to 3 ATP molecule. The second one is ATP is synthesized through complex 5. The third one is the oxidation reduction reactions produce proton gradient in respiration. Next, during aerobic respiration, role of oxygen is limited to the terminal stage. Okay, so here the option which is incorrect is 1. In fact, NADH plus H plus. So this just gives 3 ATP when it passes through the electron transport system, whereas FADH2, flavin adenine dinucleotide in the reduced form, so it just forms 2 ATP. The rest are correct. ATP synthesized through complex 5, that the ATP synthetase. Oxidation reduction reactions just calls the formation of proton gradient in respiration, the passage of protons from the intermembrane space to the mitochondrial matrix through complex 5 just leads to formation of ATP. And during aerobic respiration, the oxygen is utilized at the terminal stage in complex 4, where hydrogen and oxygen, there's the hydrogen ion, electrons, and oxygen just produce water. So the correct option is 1, because this statement is wrong. So let's now see question number 138. Plasmid PBr322 has a PST1 restriction enzyme site within the group amplifier ampicillin resistance gene that confers ampicillin resistance. If this enzyme is used for inserting a gene for beta-galactoside production and the recombinant plasmid is inserted in an E. coli strain, the transformed cell will have the ability to resist ampicillin as well as produce beta-galactoside. It will lead to lysis of host cell. It will be able to produce a novel protein with dual ability. And 4, it will not be able to confer ampicillin resistance to the host cell.
Right.
So let's now understand it with the following figure given below. So here it is the PBr322, that the plasmid of Bolivar and Rodriguez 22nd in the lab, that is used as an ideal vector to just carry the gene to E. coli. So here in this there are 2 actually antibiotic resistance site. One is the ampicillin resistance site and the other is the tetracycline resistance site. And here the restriction site PST is there. So we may just find here that this PST, it is this PST is there. PST is a restriction site that has been added in this plasmid from the bacteria. That just it is acted upon by the restriction enzyme derived from the bacteria Providencia astuarti. So the question now just says, it now just says that a gene is inserted in PST1, okay, this gene is inserted in PST1. And so what will the consequence? Let's read the question once again. Plasmid PBR322 has a PST1 restriction site within the gene ampicillin resistance that confers ampicillin resistance. That is correct. So ampicillin resistance gene here just gives a resistance to ampicillin. Okay. Now, if this enzyme is used for inserting a gene for a beta-galactoside production, and the recombinant plasmid is inserted in an E. coli strain. Okay, so suppose a function, so it will not confer ampicillin resistance to the host cell. Okay, so here the correct option is 4. So in fact, option 4 is correct, because the ampicillin gene will stop just functioning, and this is called insertional inactivation. This is called insertional inactivation.